Alphalyse is a company based on mass spectrometry. That's our main expertise, and we mainly use mass spectrometry to help clients develop new biologics. So most of our clients are preclinical or clinical phases with biologics. Biologics can be monoclonal antibodies, recombinant proteins, vaccines, and also cell and gene therapies. So what we do at Alphalyse is we have developed a method for using mass spectrometry to identify and quantify host cell proteins in those samples that we get from clients. Yes, again, back to the feature of mass spectrometry providing information about individual host cell proteins. Once you have those information, you can do a much better risk assessment. You can look at the function of those host cell proteins, and you can decide if they might be of concern related to immunogenicity or the stability of the drug itself. Things you might not discover in the early phases, but when you go into long-term stability studies, you will certainly see a problem arising with an HCP, even at a very low level. Having the mass spec data, you can do this risk assessment early on and decide if you have to optimize your process, downstream process, to actually remove specific HCPs, and then having a more safe and more stable product in the end. Yes, the identification of host cell proteins. You not only get the name of the host cell proteins, you get the PI and the molecular weight, and you can actually use those detailed information for what I call a data-driven process development, because you can use the information to optimize your process. It might be that you change the pH of a buffer or you cut a column a little bit different, but based on the information you have about the individual host cell proteins, you can do that optimization much faster. With ELISA, it's just guesswork. You would just have to set up a matrix experiments to find out how to remove certain HCPs. But with mass spectrometry, since you have the detailed information about individual host cell proteins, you can use that information to do a much faster process optimization. So it's not a requirement today by regulatory authorities to use mass spectrometry to monitor HCPs, but we have seen a development also in the regulatory environment. There's a new chapter that came out last year, USP 1132.1, actually describing best practice using mass spectrometry to characterize host cell proteins. And most people in the industry are having the understanding that over time, actually, regulatory authorities will start requiring mass spec data for monitoring host cell proteins. It's not a requirement yet, but we see more and more clients coming back saying FDA requested that we actually also provide mass spec data for characterization of our host cell proteins. Yes, I mean, of course, these very sophisticated instruments, they break down, and we have it is a production site we have here, so we have to run a certain number of samples, and the more trouble you have with either the LC or the MS, the more downtime you have, the more expensive it is to run the samples. So if we can have a more robust LC system and a very reproducible system, that makes it much easier for us to provide the services that we are providing. So robustness and reproducibility is a key factor in a setting like the one we have here at Alphalyse. Then on top of that, it's also much related to capacity. So by using the EvoSep systems, we have increased the capacity on very expensive mass spectrometry instruments. And this is of course also of value for us. If we can run 2 times or 3 times more samples with the same system, then it's of great value for us, but also for our clients, because of course then our capacity is increasing and we can provide the data faster, even for large sample projects.
EvoZap, it's a friendly instrument that it's easy to use, it's easy to troubleshoot if we need it as well with the diagnostic tests. Since EvoZap has the Evo chips, we can see that the life of the columns, they are longer than using the analytical flow as well. So we are very happy with that.
Yeah, I mean, we have run these kind of instruments for many years, and we have had different LC systems in front of our mass spec instruments, both analytical and microflow systems, also nanoflow systems. And it has been a challenge, I would say, with the other LC systems, that they break down and they are not that easy to fix, especially with the lower flow systems. It's a problem to actually identify the problems and just correct them. But with the EvoZap system, we have less problems, we have less downtime, and that increase our capacity and our production capacity, you could say. I think it's only going to increase the use of mass spectrometry for HCP characterization. It's regulatory authorities are starting to see the benefit of what you get with mass spectrometry. We have this new chapter coming out that came out last year for the USP describing best practice in using mass spectrometry for HCP characterization. And I think more and more people will now align accordingly to that chapter and start using it. So this will for sure develop into a more mainstream analysis and also being used for pretty much all projects that are related to biologics development.
Definitely. So EvoZap is delivering a great results with a good reproducibility, and also the user-friendly that they have, it helps in doing the work.
The main thing with the EvoZap system is it increases our capacity. It's a robust system that makes it higher capacity, higher or more uptime. So we can run the instruments more continuously, and that's of great value for us. It's also a system that provides very reproducible data, so we don't have to set up a different LC gradients. We just run it in one setting, and it makes it much more simple than if we had to run with different settings on our LC system. So I can highly recommend the system. It's a wonderful system, and we have had it now running for the last 3 months, and we haven't had any issues until now.